So my research has been focused on the development of scalable platforms for either expansion or neural induction of human pluripotent stem cells using suspension aggregates. So we can say that basically my whole research is based on the stem cells as a starting point. Suspension culture is very advantageous at several levels. First, the logistics. So it's easier to have just a few culture plates, 6 well plates for example, with a few wells with the cells as aggregates, than having a full incubator with dozens of tea flasks with cells attached. Also, you are saving on the cost of the matrices, so you don't need any matrices for the cells to attach to. And a third one, in using 3D cultures is really easy and it's more accessible if you really want to mimic the interactions that exist between cells in vivo. One of the main challenges that we face is to keep human pluripotent stem cells with a quality as good as possible. These cells are very, very sensitive, and small changes in their culture conditions, and even when changing the culture medium, can lead to differences in the cells. So it's important to verify that the phenotype, the karyotype, and the stability of the cells is maintained throughout the whole culture. And for that, we really need a very robust culture media that allows us to maintain those conditions. The main challenge that we face when using suspension culture is aggregation. When we seed cells either into a culture plate or a spinner flask or a bioreactor, the first step that we really need to work is the aggregation. And usually we have obtained already using different culture media, we usually have about 30% of cells that indeed aggregate. So there's a lot of cells that are lost in this process, and that's a challenge that we were able to overcome using the pluripotent StemScale. StemScale allowed us to do an every other day feeding schedule, which is very, very complying with the necessity that we have to focus on papers or grants or anything else that we may need to do outside of the lab. Another thing is homogeneity. So one of the parameters that we need to ensure that it's perfectly working in our culture is that we have aggregates roughly the same size. So if we start with aggregates that are bigger and the ones that are smaller, after 3 days we end up with very small aggregates, and at the same time we end up with aggregates so big that we start to have diffusional problems, and they may start to differentiate or even to create necrotic centers within the aggregates. What we have observed with StemScale is that we do have very homogeneous populations, and we verify that by taking some photographs each day. StemScale is indeed robust and allows us to replicate the results despite the cell line that we use. So its robustness allows us also to obtain very similar results again between passages, different passages, or even between different cell lines. There's just a few things that you need to consider just to obtain the best results that you can. So the first one is to take attention to the confluency of the adherent culture that you are starting with. So you do not want to let the cells to grow too much. You want to keep it about 60% confluency before starting to dissociate the cells for starting the suspension culture. Also consider to use a culture media that is suitable for suspension culture, such as, for example, the StemScale. Given the robustness and homogeneity that the StemScale provides to generate the suspension cultures, I think it would be important to use it, for example, to generate aggregates that can be used towards differentiation. So, the use of 3D models for disease modeling or drug discovery has been greatly increasing in the past few years, and I think that StemScale provides the quality that the cells need to initiate a good differentiation process. The quality of the cells, and especially the homogeneity of the starting aggregates, they highly depend on the outcome that we will have once we finish that differentiation. The best way to generate these organoids highly depend on the generation of 3D aggregates prior to the organization. And being in 3D will highly impact the 3D architecture of the organoids that we are trying to derive in vitro. It has been a privilege to be able to be a part of the team that has been given early access to StemScale. And it's great to be able to provide some feedback to people that are thinking of starting suspension cultures in their labs.